In this video, we will begin to discuss specific scenarios within this inheritance category. Let's begin with incomplete penetrance. This term indicates an allele that does not always show up in the phenotype, despite being present in the genotype. So in incomplete penetrance, there are instances when you can have the genotype for a trait, but not display the phenotype. For incomplete penetrance, we express the occurrence or penetrance at the population level. Thus, if a trait is 60% penetrant, that means that 60% of heterozygotes carrying the dominant allele exhibit the trait. An example of incomplete penetrance is polydactyla, which is possessing extra fingers and/or toes. Not everyone that has the genotype for this trait will express the phenotype. This range of outcomes is often due to variable expressivity of these genes. This can be apparent at the individual level, as people can express just a single extra digit, so those would have low expressivity, or have many extra digits, and that would be an example of high expressivity. But what causes this variable expressivity? To date, we recognize two possible causes, which are an environmental effect and interactions with other genes in our genomes that affect the expression of our genes of interest. We know already that the environment can play a powerful role in gene expression. And for genes that exhibit variable expressivity, the environment can definitely be the culprit. Environmental effects on gene expression can be profound. For example, temperature leads to dramatic changes in coat colors in Arctic foxes, which help them survive in snowy and non-snowy conditions. A human example is phenylketonuria, which is a mutation that makes a person unable to metabolize phenylalanine. A buildup of this amino acid can have severe consequences, and this is one reason why newborn babies are immediately tested for this condition. To avoid the severe consequences, like mental retardation, one simply needs to avoid phenylalanine in their diet. This is not actually simple to do in practice, but it is possible, and doing so allows people with the PKU genotype to avoid the severe phenotype. In addition to examples on the previous slide, we can also observe what is called the norm of reaction in response to the environment. That is a range of phenotypes seen due to environmental effects. Our example here shows that the number of facets in the fruit fly eye decreases with increasing temperature during development. This is due to temperature effects on that gene uh, that controls this trait in fruit flies. Moving on from environmental effects, we see a case where a single copy of a dominant allele is not enough to show the dominant phenotype. In incomplete dominance, heterozygotes display an intermediate phenotype between the two homozygotes. In our example here, true breeding red and white flowers combine to make pink flowers, which are the heterozygotes. When we do a monohybrid cross with incomplete dominance, we see that the phenotypic ratio matches the genotypic ratio. This is the important thing to remember here. For incomplete dominance, the genotype and phenotype ratios are the same. But what actually causes the intermediate phenotype? Well, it actually goes back to a previous topic where the amount of a protein produced by one copy of the dominant allele is not enough. Hence, in our previous slide, not enough red pigment protein results in a light red or pink color on the flower. What is remarkable is that for many traits, if we look close enough, we see evidence of incomplete dominance. For example, Mendel's pea-shaped trait actually shows incomplete dominance if we look at the peas under the microscope. It's a specific protein that makes the pea round and a lack of it that makes the pea wrinkled. In the heterozygotes, there actually is less of the protein present than in the homozygous dominant individuals. But the P still appears round and was thus counted as such in Mendel's experiments.